Hello everyone, I'm Ananth Ramakrishnan, basically a chemical engineer. I'm applying the chemical engineering principles to develop a new food product, food machinery, so on. You look at today, every day we are taking more than 1 kg of food. But how many of you are aware of that, how much amount of the nutrition you are taking every day? For simple example, proteins. Already a lot of deficiency is there in the protein among the populations. You need zero point 0.8 gram per kg of the body weight you have to take protein every day. For example, if you are a 75 kg of the weight, you need to take at least 60 gram of protein. Look at yesterday, whatever you are taking morning to evening, how much amount of the protein you have taken. Definitely it's very difficult to find out we have taken adequate protein. Apart from that, the soluble protein, digestible protein, you need to take a nutrient. If I take the blood sample of the same hall who are sitting here, definitely 30 to 40% may be anemic. That's what India's statistics also leading to anemic. How to solve this problem? How to analyze ourselves to go into that, see that how much amount of the nutrient we are taking every day. That's what today I'm going to talk about food digestion and absorption. Look at the whole population, 1 in 8 people living in obese. Not only that, 2.5 billion populations are overweight, and then 37 million population under the children, 5 years age, is the overweight. 17.9 million globally death on cardiovascular, and 1.5 million death due to the diabetes. All are interlinked with the food what you are taking every day. Earlier, we used to take food for energy, then nutrition. Now we wanted to take food for enjoyment. That's what, every day, every weekend you are going to restaurant, you are enjoying the food, you are not even looked at how much sugar, salt, fat you are taking day by day. That's what it looks like this. If you look at the India, stunting, anemia. If you go to the America, Africa, all three, stunting, anemia, and also overweight. This is continuously spreading throughout the globe. It's not only put a pressure on individual, who, you and me, it also put a pressure on the government system. The healthcare expenditures continuously has to increase. How to solve this one? You need to understand how the food is going in your GI tract. Suppose you think about now you are going to take a good biryani or globe jamun. Immediately your saliva will start coming out from your mouth without looking at the food. Then the smell of biryani will hit you in your nose. Then your eye is going to that. Then take it the biryani, putting it in the tongue, you start enjoying the biryani. Correct? That's what normally we used to do that. That's the portion of the mouth, what it does. Then it goes to the stomach. There the digestion takes place, all the food molecule disintegrate and ready for your nutrient to absorb in the small intestine. Almost it takes 1 to 5 hours in the small intestine, all the absorption takes place. Once all the nutrient absorbs in your small intestine, it moves to the large intestine. Whatever the left is removed, the water, fiber, everything, it comes out from the body. We look into the one by one. First, oral. The oral mastication is important. If you put the mouth, the tongue, it creates the state bugs. State bugs are there in the tongue. That gives a signal to your brain. Same example of biryani. You started enjoying. How much you enjoy the food, you start biting into that. For example, if you take the simple curd rice, simply you swallow it. That's the time how much is there. Then saliva comes there, that breaks down your starch molecules. Then it makes bolus form of your food, whatever you are taking into the mouth. It goes inside the stomach. This is what happening in the stomach. The contraction takes place in the wall of the stomach. And then the top portion is the fundus, and the down is antral. It's the food particles moves inside. And then the contraction takes place 1.5 mm per minute. Continuously it moves around that. It's a two-way digestion takes place. One is the first is break down your food molecules. Then the digestive juice, what is there in the pepsin and everything, yes, diffuse into your food molecules and become a smaller size particle, say 2 mm size. Then pyrolytic valve will open, it moves to your small intestine, where your digestion takes place. There, what happened is a bile salt comes out from your gallbladder, and as well as your liver solution, everything goes to the small intestine, and then absorption takes place. For example, protein will be break down to the peptine and amino acids, and also your sugar, carbohydrates converts the glucose. Then you have the fat will be break down the fatty acids, all will be absorbed through the intestinal wall. And the different mechanisms are used. One is the simple diffusion, or the facilitating diffusion, or active transport. I'm not going for detail into that, but this is what your nutrient absorbs. The larger area of the villi and microvilli, it takes the absorption of this nutrient, take into the bloodstream to the other body parts. That's what the whole absorption takes place. Now you understand how the whole process of absorption. And finally, it comes to the large intestine, is the removal of the water as well as the waste. It comes from the small intestine, and finally, it comes out from your body. The process is over. But what is happening right now? If you go to any supermarket, diabetic food, organic food, low GI food, and we really confused. Is it a really low GI food? Is antioxidants are there? Which is the food we have to select for our body? That confusion continuously going on, and how to select all these things. The same time, if you read the other side of the newspaper, people used to say, yes, these are wrong, misleading claims, and this is not the diabetic food. It's not really organic foods. Who is certifying? How it is that all these things happening in around us? As a consumer, you and me, we need to know how this has been validated. That methods are there. One method is the XYO method. Take the intestine, putting into all the nutrient, putting in the solution, see that how much is transmitted from your food molecule to the intestine. It's averted gut method, they used to say. Otherwise, we know well aware of that animals models. We can do the animal studies as well as in silico method. Different methods are there to study. Apart from that human method as well, we can go for the in vivo method, but you need ethical clearance, all these things. But even though some mistakes may happen here and there, we are using almost 115 million animals per year for testing. So huge. Second one is that sometime it will be passed in the animal, it's not cleared in the human. Best example, HIV vaccine is cleared in the animal, it's not worked in the human. There's always is a difference between the whole systemic circulations and the system itself. For example, our stomach capacity is 1.5 liter capacity, but 250 ml capacity of the rat, and all this length of the intestine, all these changes. For gallbladder is there in the human, it's not there in the animal. How you can go close to that? That is the big question mark. Can we create an artificial human digestive system? If it is there, it's a control system to study this. This is the dream for me when I joined as a CFTR as a scientist, Central Food Technology Research Institute, Mysore. I started looking into that. What are the available artificial human digestive system? These are the systems, if you look at it's available, one such system is TNO system in the Netherlands. So stomach, intestine, all these things are there. Some system has the conical back type, they given the same contraction from the outside mechanisms. I wanted to go very close to the human system. So one thing is, I want to study how the stomach inside layer, how it functions. We used to put myself, my student, we went to the hospital, we put the endoscopy, but stomach is not at all expanded. Some foreign body has come, it's not working as normally what's supposed to work. Then we moved on to look into that, what is the other alternative. One such alternative is the pig. Pig intestinal system is almost close to the human intestinal system. Look at that size. The capacity, 1 to 1.5 liter. Capacity of the human stomach, pig system comes is 3.5 to 5 liter capacity. Length of the intestine, our length is almost 6.7 meters, close to 7 meters, 21 meters. I thought of to work on that. We went to slaughterhouse, we collected the stomach, you see that is the pig stomach, and we molded, we made that silicone rubber stomach. I tried to use that, believe it or not, miserably I failed in this experiment. We lost more than 1.5 years of time what we have spent on that. Then I realized why not we can't go with the very close to the human. That time 2010, 11, 12, that time 3D printing is started coming into our nation, and I thought of to go for 3D printing, but they need a 3D image. What we did, we put the human volunteers to the MRI. MRI images comes only 2D image. So we have taken the 2D image, converted the 3D image. How it can be possible? Per minute, 120 images have taken. That means per second, 2 image. That can be considered with the 3D image, then 3D printed. This is the, I think, very early stage, we printed the stomach. This is the stomach. This is the 3D printed stomach. Almost same thing we able to recreate, including how the contraction takes place outside, internal juice and pH, everything we maintain, including the body temperature is maintained here. One important characteristic of the stomach is the emptying. When stomach is emptying, your digested material decides the nutrition release as well as your glycemic load. For example, you are a diabetic. If stomach is released, slow release in the stomach, then you can reduce your glucose level shooting up. That's the idea. Can we able to do something? Yes. If you look at water, it takes around 2 hours to release from that. Within a 10 minutes, start coming out the water. But when go for the solid foods, it takes for close to 3 to 4 hours. If a fibrous foods, it goes still higher than that. That's what you can manipulate or fabricate the novel food products. Second thing, how to open up. See, one theory is that when pyrolytic valve open, when the digestion is over pyrolytic valve, it's very difficult to understand. One study is that particle size become a 2 mm size, it will open. Another one is the calorific value in the liquid system. So we wanted to understand what we did, computational fluid dynamics modeling. We recreated the stomach, how the particles move. You can see that once viscosity is high, it moves slow. That means don't take the water taking the food. Otherwise, your glucose will shoot up as quick as possible. If soup and other things goes along with viscous material, particle movements is very less, then pyrolytic will open very late. We could able to integrate this one with our system, then the study comes. After my stay at CFTRA, I moved to NIFTM Tanjavur as a director. Along with me, the system also moved. There I tried to validate the same system what I developed. Again, we put the human volunteers in the MRI, the same time our system. We gave the apple 300 grams of apple along with 100 ml of water and the overnight fasting. We scanned the stomach of all the volunteers. You can look at that before given the feed and 0 minute to 120 minutes. 30 minutes, only 70% is there, then slowly 50% and 120 minutes, almost only 25% left. That air gap and how food particles moves, we could be able to calculate total retention time. And that time, that graph very clearly demonstrated my system, what the ARK system as well as the MRI image, both are almost similar. We could be able to recreate the stomach system. And what about to small intestinal system? That has the 3 portions I said, duodenum and jejunum and ileum, and we could able to make it the same thing. But our intestinal system is 6.7 meter, and here we made it only 3 feet. Only thing we manipulated with the residence time. And the surface area, we have to put mathematical model to account that. That's what the system, and the system also create the segmentation motion of the intestine. You can see that how the segmentation motion is created by the machine what we created in our lab. Not only that, anything if you do that food product, you need to analyze the glycemic index. How much you take after the 2 hours, how much your glucose comes to your blood. That is referred as the reference blood sample, or the maximum of 100, you can calculate it. This machine can calculate that. That is my idea to develop the mathematical model for that. But glucose response curve is entirely different like this. But our curve goes opposite. So what we did, something we missed it, that is the insulin factor. When we added the insulin factor, after 60 minutes, yes, it comes very nice. But initially it has not come. Later we realized one thing. When you are in the fasting, your body has the glucose. It is not a 0% glucose anywhere in the body. So that we able to put it, that fasting glucose or insulin, along with that, we could able to get the same curve. So this also been validated. Not only that, when you develop the product, the biscuits and glucose, Plain biscuits along with the finger millet biscuits and the biscuits with the wheat bran additions, and we could able to calculate all this GI value. You can see that human and as well as the system what it calculates almost comes very close to that. And also micronutrient also, for example, vitamin E, this is the fat soluble, that also has come very close to what we developed in this system. This is the system now moved to CSIR NIST Trivandrum. Wherever I am going, this system also traveling with me. This is the system initially that time 2010 in Asia, we are the one started working. Right now it's a full-fledged system, can calculate disintegration kinetics, absorption of the nutrient, glycemic index, glycemic load, all these things can calculate, totally developed in India itself. Very few labs have such type of system. This can sort it out lot of issues for the testing of the product. Whenever you develop the new product, you no need to go directly to the animal or human trial. You can do this system. You can go inside the things when particle disintegrate. For example, you wanted to develop the biscuit with the high fibers. You should know when particle disintegrate, when the glucose release, then accordingly you can modify. Now we are in the process of making the oral and the large intestine, wanted to attach with that very soon. That also we can able to do with the full scientific support of my students. Only thing you should know that whatever you are now, what you eat. What food you are eating, that decides your mind and your character, what you behave into that. So please understand, whenever you are taking each and every drop or each and every gram of food is going to your tongue, you should know that how much amount of the protein, how much amount of the nutrient in it. Don't think only enjoyment. Occasionally is fine, but not every meal, every 3-time meal, you should not take for only enjoyment. You should take for yourself. And you always have a good concern about good food for yourself, for the nation, and also this earth. Sustainably prepared food, always take it. Low carbon footprint processed food you should take. And we need to support for all these 3 angles. Definitely, we can make it a sustainable food system for the future. Whatever I spoken, it is written in this book. It's a combined of all food, digestion, and absorption. All details are in this book. Once again, thank you so much for listening to me.